It's my pleasure to welcome Daniel Howsmon as our speaker today for the CAST webinar. He's going to be talking today about classification, behavioral severity prediction of autism spectrum disorder using blood-based measurements. And let me just talk a little bit about some of Daniel's preparation and this award, the CAST Division David W. Smith Award. That he received, and that's why he's been invited to give this webinar today. He's currently a postdoctoral research fellow at the University of Texas at Austin, and he's developing mathematical models of valvular interstitial cell signaling to better understand how different stimuli work together to activate VICs and ultimately lead to disease. He was formerly a graduate student at Rensselaer Polytechnic Institute. He obtained his PhD in data-driven modeling of uncertain biological systems, and some of that work we're going to hear about today. He's been a summer intern at GlaxoSmithKline, Pennsylvania, and also he completed his bachelor's degree at Texas A&M University in 2012. So with that, I'll go ahead and turn it over to Daniel for his presentation. Welcome, Daniel.
Great, John. Thanks for having me. Can you see the slides okay?
Go ahead and start sharing your screen now. I think I had grabbed the screen sharing, and now you can take over.
All right.
Okay, we can see that.
Awesome.
Perfect.
Great. So thanks, John, for the introduction and for the opportunity to present today. So my name is Daniel Howsmon. I'm currently at the Odin Institute for Computational Engineering and Sciences at the University of Texas at Austin. And this work was part of my PhD dissertation done at Rensselaer Polytechnic. So as John said, we're going to be talking about classification and adaptive behavior prediction in children with autism spectrum disorder today. So before I start, I'd like to acknowledge my colleagues at Rensselaer Polytechnic. So this work was conducted under the supervision of Jürgen Hahn. I thank all my lab members for kind of insightful discussions about this, and our collaborators at the University of Arkansas for Medical Sciences, Phoenix Children's Hospital, and Arizona State University. So what is autism spectrum disorder? So autism spectrum disorder encompasses a large group of early onset neurological diseases that are characterized by two things: that's social communication and interaction difficulties, and the expression of restricted repetitive behaviors and interests. So autism, as of 2013, in the DSM-5, incorporates Asperger's disorder, childhood disintegrated disorder, and pervasive developmental disorder not otherwise specified, and autism under kind of a large umbrella. Autism spectrum disorder frequently co-occurs with intellectual disability, ADHD, speech and language delays, etc. It also frequently co-occurs with sleep disorders and GI problems, which people might not be as familiar with. And Jürgen has some other work, especially with the GI problems, that I encourage you to look into after the presentation. So in 2015, there's work done by the CDC investigating the prevalence of different kinds of developmental disorders in 8-year-olds in metropolitan Atlanta. And we see that from this chart that most developmental disorders seem to have a fairly stagnant prevalence over time in the population. However, when they looked at autism, they saw that the prevalence is greatly increasing. And so their follow-up studies in these MMWR surveillance summaries show that that trend is continuing up until now. So there's a lot of debate to why this prevalence. Is there just better, more awareness about autism in the community? Is it because of the DSM-5 criteria that like lumps these things together? regardless of the reasons why, the impact that autism has on the quality of life of the individuals, as well as their caregivers, along with these kind of prevalence increases, has prompted intense research investigations into both the etiology, pathophysiology, and diagnosis of the disease. So just to put some of the costs in perspective, current estimates of total cost in the US suggest that autism spectrum disorder costs about the same as diabetes, as well as heart disease and stroke. And you might be more familiar with diabetes and heart disease than with autism. So that just kind of lays the ground for the economic impact overall in the US. Okay, so the root cause of autism. So there have been quite a few twin studies that have been done to try to tease out the various components, whether it's genetics, the environment, whether they're rare mutations, whether they're a bunch of common mutations. And these studies suggest that most of the genetic variation is from commonly inherited traits. And this kind of corroborates with the fact that we have about 15% of autism cases have a known genetic cause. There's also a large portion that's accounted for by the environment. So these environmental effects are playing a large role in kind of the underlying causes of autism spectrum disorder. So how is autism spectrum disorder diagnosed? So currently, the diagnostic criteria are a few different behavior checklists that kind of tally a bunch of different symptoms. And in particular, there are kind of two main types of tests. One are clinical ratings after a clinician observes the child. And then other forms of assessment are from caregiver interviews, where they interview the parents and caregivers of the child. It's typically accepted that reliable diagnosis is possible after about 2 years of age. Sometimes it's possible as early as 18 months. And the American Pediatric Association recommends all children to be screened for autism between the ages of 18 months and 24 months. But most kids are diagnosed much later, and the median age is actually about 50 months. And so we know that the earlier that children are diagnosed and the earlier that we can intervene with educational and kind of social aids, they have more favorable outcomes. So the identification of biomarkers or other tools to decrease this age at diagnosis will probably lead to better outcomes for patients with autism spectrum disorder. So what is a biomarker? So I found that this is a fairly useful definition from the Biomarker Definitions Working Group, and that it's a characteristic that is objectively measured and evaluated as an indicator of a physiological process, a pathogenic process, or pharmacological responses. So typically we're talking about genetic mutations. We're talking about kind of measurements that your body's producing, metabolites, proteins, et cetera. We're talking about other kinds of physiological signals, such as brain activity or variations in ECG readings or something like that, right? We can also lump biomarkers, we can classify biomarkers as to their intended purpose, to diagnose the disease, to stratify patients, to assess different treatment options, or to assess environmental exposure. So today we're going to be looking at biomarkers that aid in diagnosis of a disease or disorder. So some of the tools that we're going to use to kind of analyze our data are different kinds of classification and regression models that are particularly well suited to low sample sizes. So in particular, we'll use Fisher discriminant analysis for classification and partial least squares for regression. So what Fisher discriminant analysis does is that if you have a labeled data set, these red points and these blue points, and we have measurements, let's say X1 and X2. So we can't draw horizontal or vertical lines on this graph to say use X1 or X2 by themselves to separate these two groups. However, what we can do is we can project down to a lower dimensional space for separation. And in particular, Fisher discriminant analysis will try to maximize the between class scatter, so maximize the distance between the red points and the blue points, and also minimize the within class scatter, so minimize the dispersion of the red points and the blue points within this space. So partial least squares is a technique that's similar to principal component analysis, but instead of just trying to explain the variation in X, we'll also try to explain the variation in a set of predictor variables Y with these techniques. We'll also extend these to nonlinear relationships via the kernel trick, where we're first going to project to a higher dimensional space where this classification is performed. And so if you see kernel Fisher discriminant analysis or kernel partial least squares, then it's a nonlinear version of these kind of techniques. So if we go back to the classification, one way that we can assess separability of two different populations is with the C-statistic. And so the C-statistic is the area under a receiver operating characteristic curve. And so what we're going to do is, if you look at this, these two distributions on the left side of the screen, we have this red distribution and this blue distribution, and we can vary this threshold in this space. And by varying the threshold, we can capture as much of this red distribution as we want at the cost of incorporating this blue distribution too. So perfect classification will have a C-statistic of 1, and a random classification will have a C-statistic of 0.5. So for this example, we might have an ROC curve that looks like this with a C-statistic of about 0.9 or so. Another thing that's really important for this kind of work with limited samples is to use cross-validation, where instead of trying to fit a model to all of our samples and assessing our errors, what we're going to do is we're going to leave one sample out at a time and fit the model to the remaining data. And with this leave one out strategy, this will allow us to use the maximal amount of data for training a model and assess, what will happen if we have a new sample into the population. So we're just going to repeat this for all of our samples. And so each one of our errors is assessed independently, so the models do not know of the sample that's left out. So this will help us ensure that we're generalizing to new data instead of just predicting the current data set. Okay, so if we go back to autism, one kind of data that we attempted to analyze for the diagnostic purposes are red blood cell fatty acid profiles. And so we read work such as that by Brigandi. In this particular study, they looked at the percentage of, say, DHA and other fatty acid profiles, and they said the mean percentage was significantly lower in autistic individuals with a p-value of 0.001, which is pretty good. And then they conclude that supplementation with DHA may be a useful intervention. So if we look at the distribution, though, of the DHA measurements between these populations, we can see that, yes, the mean looks like it might be slightly lower in the autism spectrum disorder group than the neurotypical peers, but you can see that these distributions overlap significantly. We found something similar in our own study where, yeah, the distributions overlap significantly, and there might be slight differences in the mean. And so what we found from this analysis is that, significant differences at the population level should not be equated with significant classification. And for diagnostic biomarkers, or even those that assess treatment efficacy, we should be looking at kind of separation of these distributions. So even when we try to extend this to multivariate analysis, using all of the measurements in our group, we weren't able to actually separate these populations into distinct clusters of points. So we went on and we looked at other kinds of measurements, and we saw that there are a lot of studies that looked at the association between toxic metals and autism spectrum disorder. So for the biological measurements, people will measure things like the first haircut or the first fingernail trippings of children, blood measurements or urine collection drawn at different points in time. And they usually assess these biological measurements with univariate linear regression and t-tests. Again, looking at population level differences instead of classifying individuals. There are other sets of studies that look at environmental exposure. So they'll do things like modeling your proximity to highways in the case of traffic-related air pollution, concluding that your liability for autism spectrum disorder increases closer to city centers or closer to traffic-related air pollution. So there are a bunch of these environmental exposure kind of studies too, notably in California and Israel. So we're going to look at the contribution of heavy metals in autism spectrum disorder. So in this study, we have about 120 participants, roughly split between autism spectrum disorder and neurotypical peers. We see that we have a 4-to-1 male predominance, which is about what occurs in the general population. And these are going to be the urine toxic metals that we analyze, and then we are also going to assess individuals on a number of different severity scales, including the ADOS and the CARS-2, as well as the aberrant behavior checklist. Okay, so if we use this data in a Fisher discriminant analysis, we can see that we get not great separation between the autism population and the neurotypical controls. So these are cross-validated scores and then the fitted distribution functions on the right. However, if we extend this to nonlinear interactions with kernel methods, we can see that we can actually get some separation between these two groups. Again, we have cross-validated scores on the left and the fitted model on the right. So these nonlinear interactions are important for the urine toxic elements. Similarly, if we regress onto different kinds of severity scores, the linear models, the partial least squares on the top, is not able to perform as well as the kernel methods on the bottom. So here we're using the ABC checklist as a only in the severity prediction, and then the different kind of ABC subscores. But the results hold across the other kinds of scores as well. So we need to use multivariate and possibly nonlinear techniques for this kind of analysis. Okay, so we might be onto something with the urine toxic elements. How are toxins kind of processed by the body? And so we can see the environmental toxins here are going to be removed by glutathione in this diagram. And then we said, okay, well, this folate-dependent one-carbon metabolism and transsulfuration also incorporates a number of other things that we know about autism. In particular, you can see that this MTHFR gene is one of the most highly cited as a predictor of autism risk, as mutations in this gene have been found through a number of different genome-wide association studies. We know that folate supplementation is very important for prenatal vitamins, and folate has been shown to decrease the risk for autism spectrum disorder. Finally, we have valproate over here that induces hypomethylation of DNA, and valproate is actually used in mouse models of autism spectrum disorder. So if we kind of look upstream from the environmental toxins, we can see that we can incorporate a large number of different kinds of observations that might be predictive of autism spectrum disorder risk. So in this data set that we're looking at, we have a number of different folate-dependent one-carbon metabolism, and transsulfuration metabolites. We also notice that we have kind of individual measurements like SAM and SAW, but we're also going to explicitly include some nonlinear reactions like maybe the ratios between these two things, or the percentage of oxidized glutathione. We have a slightly larger cohort in this example where we have 83 participants with autism and similar numbers of neurotypical peers. We also have 47 neurotypical siblings of those participants with autism. So these should be neurotypical, but they do share some environmental and genetic similarities with our autism cohort. So if we just look at Fisher discriminant analysis, now we're getting very good separation between these two groups, where we can see we have cross-validated scores on the left and the fitted distribution on the right. And this is just with the linear discriminant analysis. So we don't even need to extend to kernel FDA or other kinds of more exotic techniques here. So if we take a balanced approach to our misclassification error, we can see that we have less than 5% misclassification in each of these two groups. So we next looked at siblings, and we see that the sibling cohort, like I said, is more genetically similar to our autism cohort, and they also share some more environmental factors. But there's a lot more overlap with the neurotypical cohort here. And so many siblings also behave kind of behaviorally at intermediate levels between kind of a true neurotypical cohort and those actually diagnosed with autism. So this may kind of correspond to these kind of observations seen in the broader literature here. So we next did an analysis of the important variables, and we used the C-statistic to see how many variables that we should include. And we see that we are achieving near maximum at 5 variables. We left 2 additional variables in to preserve the separation between the autism cohort and their siblings. And these are the selected variables we have in our final model. And we note that we have elements of both folate-dependent one-carbon metabolism, such as DNA methylation, and transsulfuration, as seen by glutathione kind of measurements here. And here are the cross-validated scores and the separation of this kind of reduced model in this case. Here's also our confusion matrix with the cross-validated scores. So again, if we attempt to balance the misclassification, we can see that we have only 2 false positives and 3 false negatives. So our positive predictive value is above 97%, and our negative predictive value is above 96%. So this is very good separation into these 2 cohorts, less than 5% misclassification. And again, you can vary the threshold in practice if you want to weigh these different kinds of misclassifications differently. So adaptive behavior prediction, again, we attempted to look at all combinations of variables. We found that 5 variables gives us the highest cross-validated R-squared value. And our top performing models all incorporated nitrotyrosine, tyrosine, some measure of glutathione, some measure of cysteine. And these variables have been reported to be affected by high-quality vitamin supplementation that also seems to decrease autism severity in a subset of cases. So we need the nonlinear interactions to for the regression, but we only need the linear classifier for the classification problem. So this work was featured on the cover of PLOS Computational Biology, along with a little cover figure that we put together, and it was picked up by a variety of different media outlets, kind of highlighting kind of the relevance that the broader lay public saw in this work, which is really nice. It was really cool to see. So one of our collaborators had a separate cohort where they had 150 new participants with autism. They didn't have exactly the same measurements. In particular, they didn't have DNA methylation and 8-OHG that are two of our variables in our final model. So we said, okay, well, we'll go ahead and remove those variables from the analysis and retrain the classifier. And so we can see that in this model, we have a slightly different set of variables. But again, we have things like SAM and SAW that are associated with folate-dependent one-carbon metabolism. We also have things in transsulfuration like glutathione. And so we get a little bit worse separation, but still very good separation with just the Fisher discriminant analysis here. And so these are the original cohorts that we had looked at previously. And this is the new cohort of the 150 validation samples. So the classifier and the classification procedures seeming to generalize well to new data and new cohorts. And this really prompted the formation of this company called BioRosa. Feel free to check out their website, see what they're doing. They're really just trying to extend some of this work, add different kinds of measurements, more reliable ways of measuring some of these metabolites to develop a clinical test for this and really translate some of these results to the clinic. So they're actively recruiting for a clinical trial called the Metabolic Autism Prediction Study. And so we're really excited to see how this work can actually do some predictions. So they're only enrolling children between the ages of 18 months and 60 months, and they can't actually have a previous diagnosis of autism. So we're going to see, can you use this along with caregiver interviews and observations of the children to actually inform an autism diagnosis? So some of this has been repeated by other groups. So here's a group from China that has 90 participants in each group, a slightly older population. And they only have measurements related to folate-dependent one-carbon metabolism, but not transsulfuration. So they get slightly worse classification than we do, but that might be because we need aspects of both of these kind of pathways in the classifier here. So I'll also refer you to some work done by my colleague Katie, where they looked at mothers of children with either autism or typically developing children. Caveat was the mothers can't be taking a vitamin supplement, but you can see that we can actually use some of these same measurements and markers to actually separate mothers of children in these different cohorts. So there might be some evidence that we can extend this to prenatal care, which would be very, very useful, very important. So with that, I'll kind of wrap up and conclude. So we talked about the current diagnosis of autism spectrum disorders consists of caregiver interviews and observational assessments, and that diagnostic biomarkers can help decrease the age of diagnosis. We should assess biomarkers based on their ability to classify different groups rather than population level differences. Multivariate assessments and cross-validation can help achieve higher predictability. And folate-dependent one-carbon metabolism and transsulfuration may provide biomarkers predictive of autism spectrum disorder status. So with that, these are the references I have for the talk. With that, I'll take any questions.
Fantastic. Well, thank you so much, Daniel. Great presentation, and looks like a lot of work has gone into this, and it's fantastic to see the research, as you mentioned, be publicized through the, through the media and a company, and further clinical trials as well. So it shows the promise of this, and with this classification work that you've done, identifying the features and other things that are leading to better discriminating between the two populations there. I just had a question for you, or a couple of questions. With autism, you have kind of varying levels, and And what kind of work has gone into not necessarily a classification, but a regression of a level versus like a positive or negative? Can you comment on that?
Yeah. So first I'll say like the level I think is harder to assess a number towards. So I'll go back to maybe this slide here. And so with these adaptive behavior is more like the level or severity, where you might assess something like someone that is nonverbal and can't handle touch might have a lower score than someone with maybe that would be classified as having Asperger's. It might be, have some traits of autism, but still function at a high level and can hold down a job and those kinds of things. So assigning the quantitative number, I think, is very difficult because these different psychometric tools incorporate lots of different kinds of information, including, verbal, nonverbal kinds of questionnaires, but other things too, such as different kinds of GI information or sleep information or other things like that. So I think the psychometric tool on the severity, there's still some work to be done to really get a number that really reflects, your interaction with an individual. So our prediction of the adaptive behavior or autism severity in some cases is not as good as the classification, but I also think that kind of assigning a number and how these numbers change over time is hard, is very hard.
Just a follow-on question to that. The number of data points that you have, is limited because of the amount of testing that you need to do in a very controlled study, following these individuals, taking blood samples, and other samples that are in a very controlled environment to make this study effective. What kind of differences would you how would you analyze the data differently? If instead of 100 or 200 data points, if you had hundreds of thousands of data points, what kind of methods would it open it up to?
Yeah, so that's a good point. So if you did have larger numbers of measurements, so that's what we're hoping to get with BioRosa's company. It would depend on how accurately you're able to measure some of these things. In particular, some of these measurements that are oxidized or reduced forms of the same metabolite need very specialized measurements. So assuming that you can measure all of these things and you had more data points, you can use a lot of different kinds of nonlinear methods, either tree-based approaches like random forest, neural network classifiers, et cetera. Really, the sky's the limit. You could also kind of change your cross-validation strategy to be more of like a 5-fold or 10-fold cross-validation study. But really, I think the one of the main challenges here is how do you tell someone that we need to collect more data, right? So how do you tell someone like, hey, we should go collect data on tens of thousands or hundreds of thousands of participants, because that's going to cost a lot of money. So what can we do with these limited data sets to say, hey, we might have something here, we should go collect more data, and then in the future, as you start accumulating more data, then you can start to explore, whether some of these more exotic kind of techniques, give you better performance.
Fantastic. And I think, further on down the road, possible at-home test, or, if it has to be a blood sample, maybe from a clinic, something like that. What's your best, I know some of these are, urine sample might be easier to do an at-home test. But what's your thought on some of these things? Are we ever going to get to an at-home test, or is it always going to be through a clinic?
Yeah, that's a good point. So I think that we'll start out definitely as a clinic, because I think the blood-based measurements are going to be more stable, whereas the urine measurements are can be more of a function of your current exposure levels versus we think that the metabolites might be a little bit more stable. But even then, that's just going to be, so the other thing about the clinic measurements is you have to make sure that these are like fasting measurements and those kind of things, so that they're not affected by, food, just like other kinds of blood-based measurements. Where I think the at-home kind of tests are really coming in. So you're going to have some other work where they're looking at things like number of hours of sunlight, or other kinds of weather-related things, other kinds of like GI problems, et cetera, and some people with autism that live in a controlled facility. And that they are looking at, like, are you going to have a good day or bad day the next day? So you can notify the people that work at the facility, like, hey, you might need to spend a little extra time or a little less time with this person. They might need a little bit more resources or something like that. So I think for some of the at-home tests, you can start to look at things like behavior prediction over time, and that might be the best use of kind of the at-home scenarios.
Fantastic. Fantastic presentation. I'll open it up to our audience for any additional questions that you might have. You're welcome to unmute your microphone or put a question in the chat window. And just as they're coming up with those, I think it's very good results on false positives, false negatives, type 1 and type 2 errors there. I think from the standpoint of the way the medical industry works, they'd want to reduce the false negatives by quite a bit, push that threshold. And I noticed on your chart, you had about a slightly bit more, on the false negatives, 3 versus 2. But you're always talking about the where you make that cutoff. How bad would, in order to get closer to 0 false negatives, how bad would the false positives have to get to virtually, let's say you had 99.9%, in terms of accuracy for the type 2, do you have any feel for how much of an error that would introduce on false positives?
Yeah, so that's a good point. So it's hard to assess some of these, like, the tails of these kind of distributions with these kind of data sets. But from the data that I've seen in this area from our data, it seems like we would get to more like 10%, to between 10 and 15%, I'd say, misclassification error if we really wanted to have 0 false negatives and really wanted to be sure of that. Albeit this is with a limited data, so assessing the tails is hard. But you could also maybe define a window too, where you might be, how they'll do windows for things like diabetes tests and say, oh, you have pre-diabetes, which means we need to watch what you're doing more, but we're not going to say that you actually have diabetes yet, right? So you could maybe find a band in the middle where you'd say, come back in 6 months or 3 months or however, we're going to watch you more closely. And then maybe outside of that band, you could say, false positive and false negative. So I think that there is some more work to be done, and I'll see. I'd be interested to see as the data keeps coming in, where where it makes sense to actually draw those lines clinically.
Okay. Well, thank you. We had a comment in the chat. Thanks, Daniel. It was a great presentation from Javid. So thanks for that comment. I'll just echo that as well. Thanks so much, Daniel. Excellent presentation. Congratulations on your David W. Smith Award as well from the CAST division of AIChE. It's well-deserved. Appreciate you coming and presenting today as well, sharing some of this research, and best wishes on your further studies. Now you're a postdoc right now. Are you currently going for a faculty position, or what's your plan for the future?
Yes, currently looking at faculty positions. Actually have an interview tomorrow morning. So
congratulations.
Trying to move forward with those kinds of things. Yeah.
Well, we look forward to seeing additional work as you start up a research group and take some of these ideas and further explore those. So look forward to that. Okay, well, thanks everybody for joining. Appreciate you attending today, and we'll post this online.
Perfect.